All right, I'm Joshua McClure, fondly referred to as Scotch, like the drink. And I'm here to talk about another drink, ayahuasca. It comes out of the jungle. And if you've ever noticed, ayahuasca is in a bottle, it's in water, and it's not refrigerated, right? So how is it that bacteria and fungus and viruses don't take over that bottle, right? Because it's not refrigerated. It's because ayahuasca has alkaloids in it that are antimicrobial. So they're anti-infectives. They're natural anti-infective therapeutics that come out of the jungle. So this here is Tata. So Tata was a rubber tapper slave for about 20 years. And there's pictures of him after his slavery being a rubber tapper in the jungle, and he's emaciated, he's sick, he looks almost like an AIDS patient. He escaped from slavery, went back to his roots, and as a shaman, a trained shaman for this tribe in Peru. And he just recently passed away at 103 years old, right? So he was on death's door in his 20s and was able to rejuvenate himself and come back to a full leadership position, leading the tribe as a chief of the tribe. And so I want to talk about these psychedelic antimicrobials that come out of the jungle. And then I also want to talk about the psychedelic and psychoactive effects of the microbes that are actually in you right now. So for instance, 7 out of 10 of you have a herpes brain infection that is changing your personality. So you don't actually know who you are. So we'll talk about that. Are you the microbe or are you the human? So this is ayahuasca. So the yage vine right there is the one that has the alkaloids in it. The chacruna leaf has DMT in it, which also has some antimicrobial properties, but they're not as well documented. So everything I'm talking about here has been studied in journals, in mice, in top universities all over the world. This is the ayahuasca tea. It's in this type of bottle, if you go to Peru, and it's completely non-refrigerated. You don't ever have to refrigerate it. It's essentially dosed with all of these antimicrobials, and so it never goes bad. So another plant, Syrian rue, has the same alkaloids in it. So this is another ancient medicine that was practiced in the Middle East. Very similar to ayahuasca. They make a drink out of it, they smoke it. They also fill the air with it, so they burn it and they fill a room. So if there's a sick person in the room or something, they'll fill the room, and they say that it gets rid of evil spirits. Well, kind of pathogens are evil spirits, right? They're invisible. They get inside of you. They take over your mind and your personality, make you very difficult to deal with, very aggressive, cause you to attack people when you would usually be patient. It's very similar to what the ancients would call an evil spirit, right? So let's just say we could call them that too, and if science agreed with us, and called them evil spirits, it's just a word, right? Different than a microbe. So leishmaniasis here, this treats leishmaniasis, and we don't have an FDA-approved drug for it yet. So leishmaniasis is a parasite that can get underneath your skin. It's here in Florida, by the way, and it comes from sand fleas that make their home in the seaweed. And so if you've ever walked along seaweed and you see bugs are jumping out of it and they start biting you, those are the sand fleas, and they carry leishmaniasis. So do not walk on the seaweed, right? It's in the Caribbean, it's also in the Mediterranean, but mostly in the Caribbean and in many sort of health disparity countries. Syrian rue, antibacterial, antifungal, and also good for tapeworm parasite. Many of these things we don't have medications for. So those are all alkaloids that can be copied in the pharmaceutical industry. We can learn a lot from these ancient medicines. These are some of the journals that I took some of this information from. So now let's talk about some of the good things that microbes do, right? So serotonin is what causes MDMA to give you that amazing high, right? So 95% of your serotonin is produced by your regular gut microbiome. That is just feeding off of what you had for breakfast, right? So I read this and I said, is this real? This can't be real. So your normal count of microbiome is causing 95% of your serotonin. Does that mean that you could take billions of counts of bacteria and have the equivalent of an MDMA experience? I was like, no, that can't be right. So I went to the doctor and I said, what is the highest count microbiome, like probiotics, you can give me? And he said, well, there's this one for like people who have had, like, they've just had their gut cleansed, but it's way too high for you. It's like 150 billion. I said, okay, I'll take like 4 of those. So I took it home. I didn't think it was going to do anything, and I took all of it, right? I took all of it all at once. And nothing. I waited for 30 minutes, nothing. I was like, oh, I'm going to go to work. So I got in the car. And then, this song started playing on the radio, and I was like, oh, this is a really great song. I'm really loving this song. By the time I got to work, I had to hide it myself in my office because I was having a full, like, MDMA level serotonin rush into my head. I felt like my head was coming off my body. So this is true. All it was psychedelic bacteria inside of my gut causing me to freaking see the matrix underneath my desk, right? So again, who are you? So that completely changed my state. My consciousness state was completely changed by the bacteria in our gut. So there's about 4 times as many cells in your gut, foreign cells that don't have your DNA in your gut, as there are human cells in your body. I don't know if you know that. So are you the bus driver or are you the bus? Right? So microbes really matter. Whether it's a virus, a fungus, or a bacteria, they really, really matter, right? So when you take antibiotics, you really need to replenish because you could go for months just feeling blah, right? Because you don't have the serotonin production going inside of you. And you don't have to wait for antibiotics. Like, you can go take probiotics and get that high every day. What if you could just feel that way every day? Feel great every day, right? So these are the kind of bacteria that you want to go look for. You want to see a lot of those in a probiotic, the bifido, the lacto, and then this other one here I'm not even going to try to pronounce. But you want to have like 2 or 3 of each one of those in your probiotics, and you want to get up to like a billion count instead of a million count. And so those are usually refrigerated at your pharmacy or health food store or whatever, or you can order them online. Here in the United States, there's a really good brand that's very reliable that I take. It's called VSL number 3. I have nothing to do with this company, by the way, VSL number 3. So here's another thing. So the serotonin is being upregulated, obviously, and being actually produced by the bacteria. It's basically bacteria poop, right? So what happens is you get all of this serotonin being produced, but then your body is also reacting to that and creating secondary peptides. So your body is creating peptides based on what the bacteria are providing. So as a secondary sort of like manufacturing process, right? So this one, neuropeptide galanin, is thought to be involved in many critical neurobiological functions, including sleep-wake cycle. So anybody have problems with sleep cycle, right? Probably half the people in the room. It could be because your microbiome is depleted. So eating patterns and urges, so what you just feel like eating all the time is actually being controlled by your microbiome. This does not come from you. You do not have your own favorite food. Your microbiome does. Your mood, your blood pressure regulation, parental behavior, how you behave towards your children and your peers, and also neurotropic functions, which means like your level of IQ is often affected by your microbiome. So, to the dark side. Toxoplasma gondii, who's heard of this before? Okay, so this causes people to love cats. So this is when you see a cat lady and they have like 7 or 12 cats or whatever, they are for sure infected with Toxoplasma gondii. That is an irrational behavior, and if you go over to the house, you can kind of smell there's a problem, right? So this is a parasite that lives inside of cats. It takes over the mind of mice and causes the mice to produce very high levels of oxytocin whenever they see a cat. So So they literally fall in love with the thing that's about to eat them, right? So total broken heart and broken body by the time that they're going down the throat of the cat. But it can also spread to humans, especially people who have coronavirus or long COVID or a depleted immune system, and it also takes over the mind of especially pregnant women. But also, one of the really interesting aspects is the scientists who discovered this, it causes men to seek out risk, right? So extreme behaviors like jumping off of buildings, base jumping, and then also in large populations of men that are studied, often these men will just step into traffic, like not looking, and they get killed. Now, is this the Toxoplasma gondii causing them to step into traffic, to be killed, to be able to more likely eaten by a cat? We don't know. But really interesting, right? You could say that, where does the cat end? Why, is this the cat? Are you your microbiome? Is the cat its microbiome, right? Because it's coming out of the cat, causing the mouse to come to the cat. It's basically the cat is fishing. It's fishing with its own microbiome, right? It's really interesting. Now, let's talk about how this affects us, right? So infections drive aging, right? And aging drives your susceptibility to an infection. Right? So it's this cycle, right? So as you get older, you become more vulnerable to infection, and then infection actually drives the aging of your immune system, so you become more susceptible to infection, right? So, and it causes all these other things, right? So we already talked about IQ and the suppression of your production of serotonin, and so if you have pathogenic bacteria in your gut or you have yeast in your gut, it's actually suppressing the good bacteria and causing you to have not just brain fog temporarily, but also leads to dementia, depression, cognitive decline, job loss, right? Maybe relationship loss, big things in our bodies. Also causes cancers. This is all proven, by the way. Metabolic diseases, problems with your bones like osteoporosis, frailty, and cardiovascular disease. So this rapidly accelerates aging. So if there was a way that we could get rid of the bad microbiome and enhance the good microbiome, you'd have a longevity drug here. So more examples of how the microbiome is bad for you, especially HIV. We already know about this. Everybody's terrified of it. It causes all of these problems. This is a child. They look like they're 90 years old, right? It's a really good example of aging being accelerated by the microbiome, which is interesting. The HIV virus is so small that it's about 20 nanometers. The wavelength of light is about 2 or 3 times that. So it's smaller than the wavelength of light. So go back to the evil spirit, idea, right? This is a thing that has a mind, right? Quorum sensing is proven among viruses. They will all attack at the same time, so they know when there's enough of them around to attack. So they're making a decision. And this is a thing that has a theory of mind, and it lives in a world that has never seen light. It's below the wavelength of light. And so this is a thing that is like, it's living in the darkness, preying upon the beings of light. Really interesting, right? You could get a little Tolkien and Lord of the Rings with it. So herpes simplex 1, so this is the one that creates cold sores, not the sexually transmitted one, is associated with cognitive decline. 70% of the people in the room, if we're a normal population, we have a herpes infection of the brain. Anybody who gets a cold sore, it is because it is coming out of a nerve that directly attaches to the brain. So it's going back and forth between the mouth and the brain. So this is causing personality changes, and I have seen people melt down from herpes infections. It causes all kinds of things, and then ultimately it is part of death. So about 98% of people, the autopsies of dementia brains, so Alzheimer's brains, about 98% of those brains have herpes HSV1 in them. So 2/3 of the world population have H. pylori. Causes stomach cancer, colorectal cancer, and heart disease, as well as, of course, it's very famous for the ulcers in the belly. So who or what are we? Are we human, or are we just a bag full of microbes, right? Are we just the plastic bag, that holds the whatever the lunch sandwich, or are we something more than that? It's literally impossible to know. And so this is where the swarm theory of mind comes. So it's not either or, it's both, right? We are both of those things. Is the flashlight just the light bulb, or is it the battery, right? Well, you need the battery for the light to work, right? And so think of your microbiome as your battery. And so you can have better and worse batteries. We all know that, right? So what I want to do is I want to focus on getting better batteries and cleaning out the human flashlight. So the evidence suggests that the gut microbes shape neural development, brain biochemistry, our behavior, especially in relation to stress, anxiety, and depression, which are associated with aging. And so the gut microbes are key to aging. Now I'm going to talk a little bit about my company, Maxwell Biosciences, and what we're doing. We're backed by a long list of prestigious investors. And I'm going to talk about how we're affecting microbes here. So everything that I'm talking about so far is backed by science. Our drugs have been sent off to all of these institutions for testing, including DARPA actually started the funding of our program. The Department of Energy, Stanford, NYU, National Institutes of Health have done most of our big studies, and we just got a contract with the United States Army to completely take out Ebola. So our drugs are the only small molecule that is effective against Ebola, and it's 100% effective. So it may actually make Ebola something where people don't even get sick. So we have consensus on our technology. It works in animals and in the lab. I first started wondering about the microbiome and about the peptides in the bloodstream when I saw this study where these mice are conjoined together, where the skin is actually sutured together on these mice, and they share the smallest molecules of the bloodstream. And what you end up seeing is that, of course, you have an old mouse and a young mouse, and the young mouse looks old, right? So it's got high inflammation, weak antimicrobial responses, weak apoptosis, slow stem cell recruitment, weak angiogenesis, which means a very slow process of creating blood vessels, and weak skin or epithelial tissue. And you see that in the wrinkles and hair loss and that sort of thing. And so, of course, the young mouse is what we know is young, right? So guess what happens when you put the two together and they start sharing the products of the microbiome, these peptides that are in the bloodstream? You see that the old mouse gets younger, right? So they become identical to each other. So these are cloned mice. They're not having any kind of friend versus foe or sort of organ rejection, bloodstream rejection problems because they're sharing the same DNA. So you can't just go hook yourself up to your loving partner or something and get younger. So that's not a solution. So we have to figure out how is this happening, because we have the potential not just to optimize our IQ, but actually to live for maybe 1,000 years by studying exactly what's happening here, right? And this took me out of commercial real estate artificial intelligence and into biology artificial intelligence. I had to figure out what's going on here. And so we did about 2 years of blood plasma big data analysis, which is now called AI. We looked at mice, we looked at humans. The federal government helped a lot with this. Lot of fancy equipment took part in this. We came up with a list of potential peptides that are in the bloodstream. Some of these are products of the microbiome, some are secondary or tertiary results of it. LL37 is upregulated by a good microbiome. Butyrate is one of the things that does it. Butyrate was first discovered in butter, and it smells like butt, butyrate. So it's high density in your colon. So LL37 upregulated by a good, healthy microbiome. And I thought, now let's see if we could copy LL37 and give this to humans, what would happen? So LL37 is also known human cathelicidin antimicrobial peptide, and it expressed via the human CAMP gene. So we only have one of them. There's many species that have LL37 or cathelicidins, but they have many of them. Humans only have one. right? So this may be one of the reasons why elephants don't get cancer and humans do, because we only have this one immune peptide. So it's responsible for all kinds of things. It fights cancer, it fights every type of microbe and pathogen. So viruses, bacteria, and fungus are killed by LL37, even amoebas and all kinds of nasty things. It's responsible for new blood cells, new blood vessels, wound healing, inflammation management, right? So anti-inflammatory apoptosis, which is the recycling of old cells and turning them into young cells, stem cell recruitment, and of course antimicrobial and keeping your bloodstream free of any kind of pathogens. These are the various different concentrations that are in the literature of what's been found in your blood. So it cleans, soothes, heals, and protects your tissues. Now, look what it's responsible for. And now let's go back to the mouse study that took me out of commercial real estate, and do you see any similarities? It's identical, right? And so this was another confirmation for me, and I thought, okay, LL37 has got to be associated with this, right? It's not, this is not a for sure confirmation, but it's a pretty good one. So now how do you get more LL37? Good nutrition, right? Your microbiome, sunlight, so it's upregulated. The core thing that upregulates it is vitamin D, which we all know we need, right? And exercise upregulates it as well. It's also known as a heat shock protein. So saunas, the infrared light out there, all upregulate LL37. And we know that it does all of these things in animals and in humans. So it's expressed all throughout the body. Everywhere in your body it's expressed, right? So think about that. Every single tissue in your body needs this so badly that it doesn't wait on the liver or the gut or anywhere else to produce it. It's producing it itself, right? It needs it. So it's a possible superdrug. Why don't we just all take LL37? Well, it turns out that LL37 breaks down within about 6 minutes or so in some people, within 30 seconds in others. It's totally unpredictable, and it breaks down at different rates in the same person at different times. So for instance, if the person is sleeping or they're just ate or whatever, there's no statistical normalization to it. You can't do a human trial. So that was a heartbreaker for me. I thought, oh, I found it, and then, okay, this is why no one else has done anything with it, right? So I needed to come up with a new technology to stabilize LL37. I reached out to a bioengineering professor at Stanford, and she said, we can do it, and I already have the technology to do it. We're going to replace this side chain right here, which is sort of like the car keys on your car ring, right? The car keys are what do everything. The ring doesn't matter, right? So it's attached at the carbon right here, you can see, right, in a regular peptide. That's LL37. And she said, if we attach it to the nitrogen, it won't be able to break down. And so that's what we did. We attached it to the nitrogen, and it doesn't break down anymore due to the natural enzymes in your body. So we have a working mimic of LL37. That was about 5 years ago that we achieved that. Now what do we do with it? And that's about a size comparison there. So natural LL37 is there, and then we're able to mimic certain pieces of LL37, and it's about a third of the size, making it a small molecule, and it's a purely synthetic manufacturing process. So we're going after all of the bad things, right? Ebola, coronavirus, avian flu. We've already tested against them, and it works. So I'll go into the data. We're going after antibiotics. The antibiotics now have become a pollutant. They're in our river systems, they're in our lakes, aquifers, groundwater tables. It doesn't matter if you're a vegetarian, a vegan, or a carnivore, you are eating antibiotics in all of your food. It's everywhere. If you swim in the ocean, go out and swim in the ocean today, you will get antibiotics inside of you. And that means you're also getting antibiotic-resistant bacteria as you're swimming out there and eating food. They have found antibiotic-resistant bacteria and antibiotics in the deepest trenches of the ocean. They have found it meters below in the dirt of the ocean, deep, deep, deep down into the dirt of the ocean. So it's literally everywhere. So we got to get rid of this. This is a horrible contaminant. So COVID-19 and fungi are intermixed together, as well as bacteria. So I'm not going to go into detail, but COVID-19, so the SARS-CoV-2 virus loves fungi and bacteria, and it works together in an ecosystem. It creates what's called a biofilm around it so that your immune system can't get to it, and this causes something called long COVID, or the long-term effects of this SARS-CoV-2 infection. They've also shown Ebola, influenza, and other viruses. I had West Nile virus. I had it for over a year and a half, just the slow recovery from it. It was because of this biofilm. So infectious diseases today, it's a combination of this bad microbiome, right? So what you read in the literature is like this one pathogen is bad for you and we need to get rid of it, and so I'm going to prescribe you this medicine to get rid of this one pathogen. Well, what about the whole like millions of other things that are inside of you? How does that affect it, right? How does that help balance to a good total microbiome as opposed to the bad microbiome that's killing you and aging you, right? So it's not just your acute illness, remember that. Like, do you want to live a really happy, healthy life for 100 years, 200 years, 300 years? This is all possible. So Brian Johnson is going to be speaking here pretty soon, right? He's working on that, and he's actually showing it's possible. So do you want to die early or not? You have a disease, and it's called aging. So that's what we're working on. The doctor says there's nothing we can do. We've seen this over and over in COVID-19. There's another problem here. It's not just aging. It's the death of our children. It's the death of our loved ones, right? And there's nothing, they can intubate you and it actually kills you faster, right? So it's a huge problem causing sickness and death for millions and millions of people. So NIH did a study for us, and they infected Syrian hamsters, which is the gold standard. All of the vaccines and all of the therapeutics are tested on Syrian hamsters first, and then it goes into humans. And we passed with incredible accuracy. What we were able to do is actually make the sick hamsters healthier than the uninfected hamsters with our drug, right? And we think this it's because of an effect on the microbiome. It balanced out the microbiome, but we don't know. So look at the comparison here, right? So we had a similar effect on the Syrian hamsters. We're able to completely take out Ebola. We've got a potentially multi-billion-dollar contract with the U.S. Army now because of this. Just got that, so proud of it. It's incredibly potent. The drugs are at the nanomolar potency, so that's 200 parts per billion concentration. Very, very potent, and you can see it's effective against all of the coronaviruses there, right? So not just SARS-CoV-2, but SARS-1, human beta coronavirus, and then also the Middle Eastern version of the coronavirus at those concentrations. That was performed at the National Institute of Allergies and Infectious Disease. All of these other viruses are also vulnerable to our technology. You can see it's different compounds here. And we anticipate pan-virus activity, so all viruses that have an envelope, because we understand the mechanism of action and the target. So this is a study that's done on mice. We were able to save the infected mice. I'm not going to go into too much detail there. This is a list of all of the pathogens that we've tested against so far. Many different viruses. Many of these are untreatable fungus by anything else. These bacteria, these escape bacteria, so like MRSA, right, is completely shutting down the mixed martial arts sport. Like there's so many people have methicillin-resistant Staphylococcus aureus or MRSA that there's the top 10% of the entire sport is no longer available to participate. So this is a really interesting visualization. So at the top you have HSV1. And at the bottom you have SARS-CoV-2, and this is an electron microscope. Remember, these are below the wavelength of light, so they're bouncing electrons off of it instead of photons. And so you can actually see the actual physical structure of the virus, and you can see we're blowing it up. It's actually blowing up like a bomb on target. This is fully disrupted virus here. And so you can see, compare the two there. Fully disrupted looks like a bug splat on a windshield, and then you see a well-formed virus over on the left with the long spikes on it and everything, sort of the crown. This is what LL37 does, and what we're showing is that our drugs also do the same thing. It actually protects the gut, it lowers inflammation, and is able to take out bacteria and fungus. So this is another electron microscope image. That is a Staphylococcus bacteria. This is also Staphylococcus bacteria, but from from the lengthwise, and you can see what happens to it here. So we gum up the insides of the bacteria. We don't destroy the membrane, and so it's not able to communicate with the bacteria around it and say, hey, I died, and transmit resistance. So what we're seeing is we don't create any drug resistance in the bacteria, and we've tested that. So same thing, Staphylococcus over here, and you can see untreated control bacteria here, and then you can see the bacteria here. There's holes in it, and the inside looks like it's been squeezed by a fist or something. It just got squeezed up, and it's completely stopped. So it's no longer biologically active. And so this is a leakage graph here of the fluids leaking out of the bacteria. So you can see another way of confirming that it's dead. Another way of confirming that it's dead is it's not moving anymore. So that graph is showing it's not moving. So what would you need to replace the bulk of antibiotics in humans and livestock? A new mechanism of action, effective against all tested bacteria, safe, non-toxic in animal trials and human tissues, massively scalable, less expensive, and no refrigeration required. We hit all of those. So we can go to Pakistan, we can go to Africa, and it's so cheap to manufacture it that we can literally just make it cheaper than dirt, especially by distributing manufacturing, having the manufacturing done near the place where you're actually going to use it. So we compared against the top 10 antibiotics. We're much safer and much more potent than the most popular antibiotics. We've probably all taken some of these. So there's a Z-pack there. It's completely anti-inflammatory, safe in human tissues here. It is safe in mice. So we've tested at over 5,000 times the therapeutic concentration in mice and rats. This is in neurons in the brain. So we're actually able to treat brain infections because it doesn't kill neurons. It doesn't even affect them at all. And then no development of bacterial resistance at all. So there's several universities are very interested in this and are continuing to follow up. This was done by the University of Louisville. So this is how pathogens get into your brain very rapidly, right? So you have a microbiome in your nose, and some of it is good, some of it is bad. So the bad ones get it into your nasal passage and go up through the nerves, through the olfactory area, and then you'll get fungal and bacterial infections and viral infections in your brain, and it also causes inflammation in the brain, changes your personality, and you lose function, right? So function loss, depression, anxiety, memory loss, personality change, and death, these are all documented symptoms of what the doctor calls sinusitis. And so if you go into the doctor and he said it's no big deal, you have sinusitis, say, what? Like, no, it is a big deal. You need to get that out of you right away. There is a paper-thin bone that separates your sinuses from your brain. And as soon as that paper-thin bone gets eaten through just enough for a tiny little microbe to get through, that's what causes death, is a total infection of the brain coming from your sinuses. So this causes double the number of deaths that influenza does, by the way, to put that into perspective. 28,000 deaths per year. Influenza generally, on average, is about 14,000 deaths per year in the United States. So here's how we're going to be treating sinusitis. That's the first one that we're going for. It's one of the largest indications in the world. It's about a $72 billion revenue opportunity. And we'll be spraying it into the nose here. We'll have a smart device that'll be measuring patient compliance as well as asking you how you feel. It'll have a ChatGPT-powered agent inside of it. And then we plan to hit all of this function loss and restore memory, restore personality, and bring back function to people that are currently losing their jobs and their relationships to this disease. Now, it's a huge, huge indication. So in total, not just chronic sinusitis, we have the potential to address about $200 billion in about 5 years. So $200 billion of our revenue, not existing revenue. That includes veterinary indications. This is what we've got on our list here. So long COVID is one of them. I feel really passionate about this group of people that are so misunderstood by the medical community. This is our program here. So this is the schedule. We've got an absolutely incredible team that is working on this and put this plan together. Next year is the big payoff when we get done with our monkey studies, which will be done in about 6 months, and then we go into clinical safety and efficacy next year. So it's the former team from Shire. So Ed Rudnick in the middle there is the former head of R&D from Shire Pharmaceuticals. It was recently purchased by Takeda Pharmaceuticals for $64 billion. So it was his drugs that caused that much value in that company. So he's the inventor of Adderall extended release. Who here has heard of Adderall? He's the inventor of it. Claritin, and then multiple other multi-billion-dollar blockbusters that have to do with other types of diseases that you probably haven't heard of. So he also brought over the senior vice presidents with him that did that. So they're part of our team now. This is more of our schedule, has more to do with the financials. When are we going to IPO? About in 2026 or so. We're working on our Series A now, which is, if any of you are investors, we're open to discussions, and then we'll have a Series B in probably 2025, just before the IPO. We're already under contract with the U.S. Army. We have the potential for multiple billions of dollars with that contract, assuming that the efficacy against Ebola continues to be successful. We've already shown that we can suppress it. Now we have to show that we're successful in humans. We have to wait for an outbreak in Africa. Existing pharma partnerships, and then we also have partnership royalties and licensings coming in. We hope to get our first $30 million wire at the end of the month. This is what we're working on right now. I could use your help. We are in World War III and the worst economic environment in the history of anything, and I'm trying to raise $100 million at the same time. So I haven't been home in 6 months. I've been traveling, conferences, pitching constantly, because my fear is constant. It is constant. I wake up in the middle of the night screaming because I'm dealing with the people, with the militaries around the world that are telling me about what they know about what bioweapons the terrorists are creating right now. And we can defeat them. So if you have any motivation about this sort of thing, I really hope that you'll help me preserve the civilization against what these agents of chaos are trying to create. Thank you so much. My name's Scotch McClure. That's my personal cell phone number on there, and that's our Director of Investor Relations right there if you want to talk. Happy to take any questions, anything at all. Okay, so he said he's curious about the Syrian hamster study and the weight gain that was shown above the baseline. Does that have any effect, or did the microbiome change affect them, or is there something else that caused it? We don't know. So what we saw was that we saw weight gain. The weight gain was caused by they were eating more. So they were hungry and they wanted to eat more, and the sick ones were curled up on the bottom of the cage, suffering from COVID, right? And so we don't know why, but we think that they were eating more because they were healthier, right? And so they actually gained more weight than the uninfected hamsters, which is interesting, right? That could just be a rebound effect where they were sick and then they got better and they're like, oh, it's great to feel good again, right? We know that feeling. Any other questions?
We only have time for one more.
Okay.
Question.
Yeah. So she said, is the dosing different in the acute phase of a disease versus the chronic phase of a disease? And that would be totally up to a doctor and your own personal experience of a disease. So we have not done any animal studies on any chronic diseases because it's like, imagine how expensive that would be to go for years watching a rat or something. Yes. So we're designing this as a cure. We're an anti-pharma company, right? So we're actually, the vision of the company is to create health for the world safely and affordably, right? And so, like, anybody have an iPhone in your pocket, right? So this is the most advanced communication technology in the history of the world, and each of us is able to afford it, right? So investors come to me and say, like, how can you make a profitable company when your goal is to make medicine affordable? And I just say, do you have an iPhone in your pocket? The most profitable companies in the world, the most powerful companies in the world, make affordable technologies. It's completely opposite of the idea that is dominating and the financial incentive system that is dominating pharma and breaking the bank completely. Medicare, Medicaid are failing. The doctors and nurses in the United Kingdom are striking right now. They're not going to work because the system cannot afford to pay them because they're paying for so many pharmaceuticals from the United States. It's broken. It's completely, not only is it not sustainable, but it's breaking now. It's unsustainable this year. It's failing completely. So the timing is perfect. We're gonna completely rewrite what's going on. So he's talking. Go for it. Right, so what additional indications do we plan on going after? So that's why we're raising $100 million. So we could do the full human trials and everything for about 20 million. We're raising about 5 times that to expand the AI side. That's my background, right? And to expand out and to look at what are the other things we can do. So because the nasal spray gets about 30% of the molecule that leaves that nozzle into the brain. We've seen that in the rats, right? And so we want to go after, for instance, like infectious dementia, and encephalitis, and all of these indications that there is no drug for right now. So any kind of viral indication, like viral encephalitis, meningeal encephalitis is what I had when I had West Nile virus. They said there's nothing we can do. Keep him hydrated, and then they would just send me home when I would go to the ER. And I lost my job, I lost my family, I nearly lost my life, and I was in a coma for about 2 weeks. Because of that. There's no drug for it. So we want to go after the things that there aren't any drugs for right now. So it's not really a disruption of current existing medicine. It's a disruption because it's a new medicine for things that are not treatable. It's a disruption of the underlying financial incentives, right? We want to produce this because it's one drug for so many different bugs. You can scale this one drug to be more scaled than any other drug in human history. So it's, we believe, bigger than the invention of antibiotics. Thank you, I gotta go.